So like eukaryotic cells. The ribosomes, these are non-membrane bound organelles found in all cells, like both eukaryotic and prokaryotic. Ribosomes also found in two organelles, in chloroplast in plants and in mitochondria and on rough endoplasmic reticulum. Now, animal cells contain non-membrane bound organelle. It is centrosome. Centrosome made up of two cylindrical centrioles which lie perpendicular to that helps in cell division. Like mycoplasm, the smallest cells, 0.3 micrometer in length, while bacteria is 3 to 5 micrometer. Now ostrich, an isolated single cell, the largest one. now Human RBC is 7 micrometer in diameter, and nerve cells, these are the longest cells. And so it varies, like cell shape varies with the functions they perform, right? now Nerve cells, these are branch and long, tracheid, it is elongated. Mesophyll cells, they are round and oval. Columnar epithelium cells, these are long and narrow. White blood cells, these are amoeboid. Red blood cells, round and biconcave. Now prokaryotic cells, represented by bacteria, blue green algae, mycoplasma and pp yellow, pleuronumina like organisms. Okay, these are smaller and they multiply more rapidly than the eukaryotic cells, right? Now, four basic shapes of bacteria are bacillus, that is rod shaped, coccus, that is spherical, vibrio common shaped, and spirillum, that is spiral. Now prokaryotic cells and almost similar, prokaryotes have a cell wall surrounding the cell membrane except in plasma, because now no well-defined nucleus in prokaryotic cells. Now they are basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, the prokaryotic have single chromosome circular DNA, right? And circular DNA, right? Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called, these are plasmids, right? Now this plasmid DNA contains certain unique phenotypic characters to such bacteria. Okay. uh, Now many bacteria have small circular DNA outside the genomic DNA. Now these smaller DNA are called plasmids. Now this plasmid confounds the unique phenotypic characters to such bacteria. Now one such character is resistant to antibiotics, right? Now the plasmid DNA is used to monitor bacterial transportation with foreign DNA, right? Now nuclear membrane is found in eukaryotes, right? No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Now prokaryotes have something unique in the form of inclusions. Now specialized differentiated form of cell membrane called mesosomes is also characteristic of prokaryotes. Now enfoldings of cell membrane, right? Now prokaryotes have inclusion bodies. These are like gas vacuoles, right? Now cell envelope and its modification. Just come to the point, like there is gram positive bacteria, gram negative bacteria, uh, um, based on the cell envelopes, okay, or cell wall, you can say. And then glycocalyx, it is layer over the cell wall, and it can be slim layer or capsule is tough, slim layer is loose sheath. Okay. Now cell wall determines the shape of the cell and provides a structural support to prevent the bacterium from bursting or collapsing. Now plasma membrane is selectively permeable. Okay. It is structurally similar as eukaryotes. Now mesosome, these are extensions of plasma membrane. Now this can be in the form of vesicles, tubules and lamella. Now they help in cell wall formation, DNA replication, DNA replication, distribution to daughter cells, and respiration, also secretion process to increase the surface area of the plasma membrane and enzymatic content. Now some prokaryotes like cyanobacteria, there are chromatophores that has pigments. Okay. They are also the membrane extensions into the cytoplasm. Now bacterial cells can be motile or non-motile. If motile, they have the flagella, and the flagella has three parts: filament, hook, and basal body. Now the filament, you see now the there is pili and fimbria, though both are pili for conjugation, for reproduction type of thing, and fimbria for attachment, right? Now fimbria, it is bristol, small bristle like fibers, and pili, these are elongated tubular structures and made up of proteins, right? Now in some bacteria, this fimbria attach the bacteria to rocks in streams and also to the host tissues, and pili and fimbria do not play a role in motility, right? Now ribosomes and inclusion bodies. So in prokaryotes, ribosomes are associated with the plasma membrane of the cell. Okay, now ribosomes, these are 70s in prokaryotic ribosomes. Now these are site of protein synthesis. Now several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. Now the ribosomes of a polysome translate the mRNA into proteins. Now inclusion bodies, it is a unique in prokaryotes. Now inclusion bodies, these are reserve material. These are reserve material in the cytoplasm and not bound by any membrane. They lie free in cytoplasm. These are phosphate granules, cyanophysin granules, and glycogen granules. Now another inclusion body, these are gas vacuoles. Okay, now so another inclusion bodies, these are gas vacuoles found in prokaryotes, in blue green and purple green photosynthetic bacteria. Now eukaryotic cells, now it includes all protist, plants, animals, and fungi. Now they have nucleus with a nuclear envelope, and eukaryotes have like complex locomotory and cytoskeletal structures, and genetic material organized into chromosomes, right? Now all eukaryotic cells are not identical, okay, because plants have cell walls, plastids, and large central vacuole absent in animal cells, and animal cells have centrioles which absent in almost all plant cells. You see the peroxisomes present in both animal cell and plant cell, and then there is ribosomes in the cytoplasm. In here as well, ribosomes on, okay, or ribosomes on the endoplasmic reticulum, that is derived from the nuclear envelope, right? Rough endoplasmic reticulum. Now cell membrane, talking about the RBC membrane, is mainly composed of lipids and proteins, but also have the carbohydrates and cholesterol as well, and lipids and proteins. So the lipid content is 40%, and protein content is 52%, and the layer is lipid bilayer. Now the major lipids are phospholipids, okay, and now in which polar heads towards the outer sides, and that is polar heads, that is hydrophilic polar heads, and hydrophobic tail towards the inner part, okay. And you see in the diagram, like, now depending on the ease of extraction, membrane proteins can be classified as an integral and peripheral. Now peripheral proteins lie on the surface of the membrane, while the integral proteins, they can be partially or totally buried in the membrane. Now you see there is peripheral protein, then on that there is sugar, that is maybe carbohydrate, as I said, and there is phospholipid bilayer, then integral membrane, then cholesterol. This is a fluid mosaic model of plasma membrane, right? An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. Now according to this quasi-fluid nature of lipid, now The nature of the lipid is quasi-fluid. It means it enables the lateral movement of proteins within the overall bilayer. Now this ability to move within the membrane is termed as its fluidity. So bilayer lipid has the fluidity that makes proteins to move laterally. Okay. And then there is plasma membrane is selectively permeable. Now it shows passive transport, osmosis, and active transport. Osmosis from higher to lower by water, and active transport sodium and potassium pump, and passive transport like without loss of energy, same higher to lower. And example is neutral solids. Okay, neutral solids. Now cell wall. Talking about cell wall, it is non-living rigid structure. It's not living, and present in fungi, that is chitin, and plants, that is cellulose. Cell wall not only gives a shape to the cell and protects the cell from mechanical damage and infection, helps cell to cell interaction, and provides barrier to undesirable macromolecules. Okay, and cell wall is completely permeable. Algae cell wall, cellulose, galactans, mannans, and minerals like calcium carbonate, and plants cellulose, hemicellulose, pectins, and proteins. Now young plant cell have primary cell wall that capable of growth. They gradually diminish, and the secondary cell wall is formed on the inner side, like towards the membrane side of the cell, right? It means like uh, outside is cell wall, then primary cell wall, then inside secondary cell wall, and then inside this plasma membrane. So this is how it. Now middle lamina mainly made up of calcium pectate. What it does, holds or glues the different neighboring cells together. Now cell wall and middle lamella are connected by plasmodesmata, which connects the cytoplasm of neighboring cell. Now plasmata, these are cytoplasmic threads that connects the neighboring cells, and this plasmodesmata is made by cell wall and the middle lamella. Okay, now endomembrane system.